All right, welcome. If you're a clinician, you've probably noticed something. Genetic reports are showing up on your desk more and more. And let's be honest, figuring out what they mean can be a lot. So today, we're going to demystify the whole thing. We're going to break down the gold standard framework for classifying variants so you can read those reports with confidence. Ready? Let's get into it.
So you've been there, right? You order a genetic panel, maybe even a whole exome for a patient, and what comes back? A report just filled with sequence variants. And the big question, the one that really matters, is which one of these is the answer? Is any of them the answer? Nailing that down is, well, it's everything for your patient's care. And it's a challenge that's only getting bigger for all of us.
You know, for a long time, it was kind of the Wild West. The language labs used was all over the place. You'd see terms like mutation or polymorphism thrown around, but they were super loaded terms. One person's mutation was another's polymorphism, and that led to a ton of confusion. You could have two different labs looking at the exact same variant and describing it in totally different ways. Talk about a nightmare for clinical consistency.
Okay, so something had to change. And it did. The big players, the American College of Medical Genetics and Genomics, that's the ACMG, and the Association for Molecular Pathology, or AMP, they teamed up. They saw this problem and said, we need to fix this. So back in 2015, they put out this landmark joint recommendation. The goal was simple but massive: create one single evidence-based system so that everyone, every lab, every clinician was finally speaking the same language.
And honestly, the best way to think about it is to compare it to a pathology report. You get a path report back, you see the word malignant or benign, and you know exactly what that means. There's no ambiguity. You expect that from any lab in the country. Well, that was the whole idea here, to bring that exact same level of clarity, that same consistent, reliable terminology into the world of genetic reports.
So what did they come up with? At the heart of this whole thing is a standardized 5-tier system. That's it. They created just 5 specific terms that now form the common language for every single genetic report. If you can understand these 5 categories, you've got the key to interpreting pretty much any report that comes across your desk. Yep, just 5. That's the magic number. I mean, think about the insane complexity of the human genome, right? And this framework boils it all down, all the clinical interpretation for any variant, into just one of these 5 buckets. It's pretty brilliant. So let's break them down, one by one. First up, pathogenic. This is as clear as it gets. When you see pathogenic, it means the evidence is just rock solid. We're talking well-established, published data, functional studies, the whole shebang. It all points to one thing: this variant causes disease. For you, this is a confident, clinically actionable result. Full stop. Okay, next we have likely pathogenic. Now, the word likely can feel a bit wishy-washy, but here it's actually very specific. This is one of the coolest parts of the guidelines. They put a number on it. Likely pathogenic means there's a greater than 90% certainty that this variant causes disease. So yeah, it's not 100%, but it's really, really close. You can act on this with a very high degree of confidence. All right, now for the big one. The one you're gonna see a lot and the one that causes the most headaches. The variant of uncertain significance, or VUS. Now, listen closely, because this is critical. A VUS does not mean maybe it's bad. It's not a shrug. It is a very precise statement that says, right now, we don't have enough evidence to call this one way or the other. The data might be weak, it might be conflicting, but the bottom line is, the jury is out. It's an inconclusive result, period. Flipping over to the other side of the coin, we've got likely benign. And just like likely pathogenic, this isn't a guess. It's anchored to that same standard, a greater than 90% certainty. So, the evidence is pointing very strongly to this variant being harmless. There's just that tiny little bit of uncertainty left that keeps it from being called definitively benign. And last but not least, benign. This is your definitive all clear. The evidence is strong, it's consistent, and the lab is confident in saying this variant is not the cause of your patient's problem. You can safely rule it out and move on. So you might be wondering, okay, how does a lab actually decide which of these 5 boxes to put a variant in? Is it just a gut feeling? Absolutely not. It's a super systematic process. Think of it like a point system.
They gather all these different lines of evidence, and each piece of evidence has a specific code and weight, all laid out in the guidelines.
And this evidence comes from a few major buckets. They look at population data, basically asking, is this variant super common in the general population? Because if it is, it's unlikely to cause a rare disease. They use computational data, computer algorithms that predict if a change is likely to be damaging. There's functional data from lab experiments that directly test, does this variant actually break the protein? And then there's segregation data, which is huge. That's where they look at a family and ask, does this variant actually track with the disease from one generation to the next?
Okay, we've covered the what and the how. But now let's get to the most important part, the real reason we're here. What do you, the clinician, do with this information? How do you translate one of these reports into actual clinical decisions for your patient?
All right, if you remember one thing from this entire explainer, make it this. It's a direct quote from the guidelines. A variant of uncertain significance should not be used in clinical decision-making. Let me say that again. Do not use a VUS to make clinical decisions. That means you don't change medications, you don't alter screening or surveillance protocols, you don't guide reproductive decisions based on a VUS. A VUS is a flag for, we need more information. It is not a call to action.
So when that report with a VUS lands on your desk, what's the game plan? It's pretty simple. Step 1, do not change clinical management. We just covered that, but it's worth repeating. Step 2, start the process of trying to get it reclassified. This usually means working with the lab or a genetic counselor. And you know what the most powerful tool is? Testing other affected and unaffected family members. That's called segregation analysis, and it can be a game changer. And step 3, you can of course consider monitoring your patient based on their overall clinical picture and family history, but you're doing that based on them, not based on the VUS.
This table really brings it all home, doesn't it? It's a perfect summary. If you see pathogenic or likely pathogenic, yes, you can use that for clinical decision-making. For likely pathogenic, you'll feel even better if it fits with the rest of the clinical picture, but it's still actionable. Uncertain significance? We've said it a dozen times, but here it is again. Do not use it for clinical decisions. And if it's likely benign or benign, you can breathe easy and conclude that this specific variant isn't the problem.
And one last, really crucial point. This whole process, it's a partnership. It's a two-way street. The lab brings the deep genetic expertise, but their analysis is only as good as the clinical information you provide. A detailed patient phenotype, a thorough family history. That information is gold. It gives the lab the context they need to make the right call. It truly is a collaboration between the clinic and the lab.
So, at the end of the day, what this framework does is replace confusion and ambiguity with a clear, shared, evidence-based language. It's designed to empower you, to make you more confident when you interpret a report and when you talk to your patients. And that brings me to a final question for you to think about. Now that you have these clear definitions, how is this going to change the conversations you have with your patients about what their genetic test results actually mean for them? Thanks for joining me.